Thank you for joining me today at this Interacoustics Academy webinar. As the title suggests, I'm going to cover a mix of topics that hopefully bridge the gap between some of the theory and the practical aspects of performing good otoacoustic emission measurements. Now, just before we dive in, if you would just permit me to briefly introduce myself. So, I spent 5 years on clinical otoacoustic emission research starting in 1984 at the Institute of Laryngology and Otology, working under Professor David Kemp. And I then spent 25 years in the industry performing R&D activities, working on otoacoustic emission equipment. And then approximately 6 years ago, I moved to the Demont Group, and I work closely with Interacoustics and mainly on otoacoustic emission and ABR screening products. So what are we going to discuss today? So the webinar is divided into what I hope are 3 learning outcomes. And as in my previous webinar, I've tried to address topics that frequently appear in our email inboxes or from conversations that we have with audiologists in the clinic. And the first of these addresses the fundamental differences between the types of otoacoustic emissions that we measure. And I'm going to address this from a slightly different angle to normal, I think. Next, I want to talk about the limitations of measuring DPs at high frequencies, particularly in the adult population, and what we should be mindful of as we try and interpret these results. And finally, just a brief topic looking at the influence of noise in our emission measurements and what we can do at a practical level to improve things. But before we start that, I want to just give a very quick kind of hand-waving overview of otoacoustic emissions and start by saying it's an active process that exists in the cochlea that provides an enhancement to our hearing. And this enhancement provides both an improvement in the hearing threshold and an increased frequency selectivity. And more specifically, what that frequency selectivity does is it enables us to resolve closely spaced frequencies more easily, which is one of the factors that gives us the ability to understand speech in noise. Now, our understanding is that it's these hair cells, specifically the 3 rows of outer hair cells, that contribute to this enhancement in hearing. As the acoustic travelling wave passes along the basilar membrane, these outer hair cells impart some energy into the wave by employing micromechanical movements of the cells, and this energy provides a kind of signal amplification. Now, luckily, we're able to observe the presence of this energy that's been added by the cochlea because of the very, very small amount of it that arrives back in the ear canal as acoustic energy. However, the challenge is, to be honest, what is the most reliable way to measure this energy? And finding solutions to that's kind of kept me interested in this field for pretty much my entire working life. Now, this slide's just a quick reminder of what the cochlea looks like once it's been subjected to too much sound. And many experiments have shown that as the outer hair cells diminish in number, the otoacoustic emission reduces and eventually disappears. And along with it goes our good hearing threshold and our ability to understand speech in noisy environments. What I would like to do then is to look at the various phenotypes. So we have a baffling number of acronyms that are used to describe otoacoustic emissions, and I'd like to just kind of go through what the various types are and how we've come to arrive at those. And broadly, we can categorize them into spontaneous emissions and stimulated emissions. And some people have spontaneous emissions, which can be measured simply by putting a microphone in the ear canal. But clinically, this is not proven to be very interesting. We call these type SOAE, spontaneous otoacoustic emissions. And however, we're going to be discussing emissions that are evoked using a stimulus. So for now, we'll leave the SOAEs behind. Our probe will have a loudspeaker as well as a microphone. And the stimulated emissions, they are generated as a result of the energy input by the outer hair cells in response to a sound that has been presented to the cochlea. And it's then measured by the microphone in that probe labeled 2. As you can see, Professor Kemp very neatly encapsulates the phenomena that we're studying here, and that it's pure luck that the emissions are measurable in the ear canal, and fortunately that measurement gives us insight into the fact that these outer hair cells are adding energy to the system. It serves no purpose the fact that some of that energy escapes, but luckily for us, it does give us a clear indication that the cochlea is working as it should. And if they're not present, then we know that the thresholds are impaired, and that the amplification that we will require, we're now looking at a sensory neural hearing loss, and the amplification that is required will need to use some compression in order to fit the entire dynamic range of sound into the remaining available hearing range. So now we're going to talk about the various types of stimulated otoacoustic emissions, and what they all have in common is that sound goes in, energy is added by the outer hair cells, some of this energy escapes, and it's measurable in the ear canal. Now, rather than try and explain the differences and the similarities of these 3 types of emission from a cochlear mechanics standpoint, I propose to look at this classification in relation to the evolution of robust techniques developed to measure this rather small signal. And so we're going to look at 3 different types. There's the stimulus frequency emission, the transient evoked emission, and the distortion product emission. So let's introduce our stimulus signal into the cochlea. So in this instance, it's going to be a 2 kilohertz sine wave. And the energy that is contributed by the outer hair cells that increases the amplitude of the travelling wave as it moves along the basilar membrane, this amplification is present at the same frequency as the stimulus. So in other words, energy is added at the same frequency. Therefore, when the energy escapes back out to the middle ear, it is also at the same frequency as the input. So we have a microphone that is going to measure something in the ear canal, and obviously the stimulus is still present in the ear canal, but the otoacoustic emission has been added to that stimulus. And the problem that presents itself is that the emission is probably around 1,000 times smaller than the stimulus. And so when we see or record that waveform in the ear canal, it's very difficult for us to know whether it's about 0.1% bigger than what we actually put in. And obviously, if we're trying to detect whether the cochlea is producing an emission, it's not very easy to be certain when there's such a small change in the signal that we're measuring. Now, there are tricks around this. You can slowly change the frequency. It's pretty complex, and for now, it's not a regularly used clinical tool. It's more of a research process. Now, this type of emission is called a stimulus frequency emission because it is at the same frequency as the stimulus. And in some ways, that's a little bit misleading because a transient emission is also a stimulus frequency emission, but we'll come on to that shortly. So what can we do to make a more reliable measurement of this emission? And this diagram shows actually one of the tricks. Now, fortunately, it's a non-intuitive property of the cochlea, but the travelling wave actually travels more slowly in the cochlea than you would imagine. So normally sound travels at about 30 centimeters in a millisecond. Now, in the cochlea, after 1 millisecond, the travelling wave has barely started its journey to the apex. And so this slowing down of the sound in the ear canal gives us the opportunity to measure the emission after a surprisingly long period of time. So we can present a stimulus, and then we can stop the stimulus temporarily and measure the response in the silence between the stimuli. Now, this diagram actually is an indication of how people use ultrasound to measure, image the body, and they've got the same problem, which is that they are looking for a very faint reflection from the interfaces between organs inside the body. And so they use the same technique. They basically send in a stimulus and pause and listen. Let's just represent this now in a slightly more simplified manner. So we're going to take the cochlea and our microphone and loudspeaker and just present them as a simple triangle with the basal end of the cochlea at the left-hand side. We're going to introduce a loudspeaker and a microphone to measure the response. So we're going to apply a repeating stimulus to the cochlea, and we're going to listen in the gaps between those stimuli. And in this case, we're going to apply a rectangular pulse. So the membrane is going to add energy, the basilar membrane, is going to add energy as that pulse travels along. And what you'll notice about this diagram is that the pulse starts out being extremely sharp, rectangular, and that's because it contains high frequencies as well as low frequencies. And as it moves along the basilar membrane, those high frequencies are absorbed away. So the time it gets to the apex of the cochlea, it's only the low frequencies that remain. And that frequency-specific nature of the cochlea in terms of adding energy, it also absorbs the energy. So we know that the high-frequency sounds cannot propagate beyond the basal end. So that's why the pulse shape is being modified as it travels along the membrane. But we also know that the energy that is being added is frequency-specific. So now we can look at what the energy added looks like. And so this is the response of the hair cells as that pulse moves along the cochlea. That's what we measure in the ear canal, but obviously in normal circumstances the energy doesn't wait in the cochlea until the pulse has got to the end, so this would be more representative of actually what happens. And this slide teaches us something, which is that the high frequencies start to come out whilst the pulse is still travelling along the basilar membrane, and that we mustn't send the next pulse in too quickly because we would not be able to see the low frequency emission, and also that the high frequencies come out so soon after the pulse that we have to be very careful when measuring the high frequency transient emissions that they're not contaminated by any residual energy that remains from the large transient stimulus. So this is a real one that's been recorded, and at the beginning we have had to insert some silence just to blank out the residual stimulus, because the stimulus is around 1,000 times bigger than the response. And there's the response with the high frequencies at the beginning and the low frequencies later on. So I hope that does kind of convince you that actually a transient emission is a stimulus frequency emission, because it's responding at the frequencies of the stimulus. And the reason that we call it transient is because we just quite simply can't easily measure the response at the same time as the stimulus. So we have to break the stimulus up into sections and just listen for the response in the silences. So we've separated out the stimulus from the response using time. Now, the next part is to look at distortion products. And before I do that, I'd like to just introduce the concept of distortion and the way that it's easy to talk about that using a different representation of the energy. So up until now, we've been looking at waveforms. And a waveform is a representation of pressure versus time. So the horizontal axis is time and the vertical axis is pressure. And we can equally represent that data using a frequency spectrum. And a frequency spectrum has frequency along the horizontal axis and amplitude on the vertical axis. And the red bar represents exactly the same information as the blue waveform, because if you look at the frequency axis, it shows 5 for that bar, which is exactly the number of waves in the waveform, and the amplitude shown by the height of the bar is the amplitude of the wave. Now, you'll see later why looking at things in the frequency domain is easier in this context. Now, imagine that pressure waveform is being produced by a loudspeaker cone that's moving backwards and forwards, and the loudspeaker gets damaged in some way, dropped on the floor, and so that now the cone can't move freely in both directions. That would now produce, for example, the green waveform there, and that has got a little bit of clipping at the bottom of each wave where the loudspeaker cone is restricted in its movement. And as that waveform moves along the basilar membrane, it would be detected at the characteristic place. The outer hair cells would be adding energy and the inner hair cells would be detecting the presence of that waveform. Now, there's no way that the hair cells can actually detect that clipping at that place in the cochlea. I mean, they're very sensitive, but they would not be able to determine that the waveform had been a little bit flattened. So actually, if we look at what the frequency spectrum of that waveform is, you'll find that it actually looks like that. And so that gives us the clue as to why it is we can hear distortion so clearly. And distortion is it's the redistribution of energy from the frequency that we intended to other frequencies. Okay, so in this case, because it's a single sine wave and we've distorted it, we've got energy now at the second, third, fourth and fifth harmonics of that waveform, and that is what our ear hears when we hear distortion. So that would not sound too bad actually, but if you're listening to music through a distorted loudspeaker, it's the redistribution of all of the intended frequencies to frequencies that we didn't want to hear that makes distortion sound so unpleasant. And we can use the same thing to measure the distortion in the amplification process inside the cochlea, because when you add energy, invariably there's a small amount of imperfection in the system, and that amplification process introduces some distortion. And so if we look at this in the context of a distortion product measurement in the cochlea, so again we're going to have frequency on the horizontal axis, energy or amplitude on the vertical axis, and this is a measurement from my ear using two tones, fairly closely spaced, and we refer to those as F1 and F2. And that is not the whole picture, because of the imperfections in the amplification process, where those two tones were present, my ear actually introduced some distortion. And so the blue lines have been generated by my cochlea, and they are all mathematically related to the frequencies of the tones that we put in. They're intermodulation distortions. And clinically, we look at 2F1 minus F2, as that's the one that's most robust clinically. And although you don't often see a diagram in that detail, what we're more used to looking to is a picture like this. And that is because we've performed multiple measurements to obtain that picture. So we've performed the DP measurement at different frequencies, and that graph shows the height of the DP, the 2F1 minus F2 component, and the noise in grey, which is derived from the red bars in the large graph. So looking a little more detail at what's happening in the cochlea during this process of distortion generation, let's just here's a diagram that just represents the travelling wave going along the basilar membrane, and I'm going to take that concept and simplify it. So now we have the base of the cochlea on the left apex on the right, and this is the tone of F2 being presented along the basilar membrane. And the outer hair cells have been adding their energy as it travels along, and the signal builds to a peak at its characteristic place. And I've marked a dotted line around the waveform to show the envelope of the amplitude. And then we're going to introduce a second tone, which is lower in frequency. And you can see that both of these tones cannot travel beyond their characteristic place. So the energy is absorbed once it gets there. So the high frequencies cannot travel as far. So F2 cannot travel to the F1 place because F1 is lower in frequency. Now, the DP, the 2F1 minus F2 component, requires both tones to be present in order to generate that distortion. So we know that somewhere around here some magic happens and the distortion is created and it travels back to the middle ear, to the ear canal where we measure it. Now those amplitude envelopes allow us to make some estimate of where that distortion is created, because it's most likely to be created where the maximum amount of energy of both F1 and F2 are present. And so we can multiply those two envelopes together and create a picture of where the most amount of combined energy is present. And so I've done that with this red envelope. And you can see clearly there that the most likely place of DP generation is actually very close to the F2 place in the cochlea. Also, what you can see at that point is that at the DP generating point, there is more F2 present than there is F1. And that's because you can see from the envelopes, both of the stimuli were at the same level. And so actually to maximize the response for a given amount of stimulus energy, it makes more sense to have a slightly smaller F2 than F1. And that's why in the clinical situation, we typically use an F2 with a lower stimulus intensity than F1. And that kind of balances the amount of energy in both stimuli at the site of generation. Now there's one other thing to consider that we can explain with this diagram, which is that the distortion product is at a lower frequency than F1. Therefore, when it is generated, it will travel further up the cochlea to its specific place, because that'll be further towards the apex. So this diagram shows that we have a DP that's been generated at the generating site, and it's moved a little bit more towards the apex, where it has received some amplification from the outer hair cells. And you can think of this as like another stimulus frequency emission, because as it meets its characteristic place, some of that energy will get sent back down the cochlea. And so now we have a situation where the site of primary generation is sending energy back, and the characteristic place of the DP frequency is sending energy back. And so that travels back to the middle ear and mixes with the first component. And that can either add to or subtract from, depending upon the phase, and contributes to what we call the fine structure of DPs. And the other webinar earlier discusses that in more detail. So to summarize, we've got 3 different types of stimulated emissions, and the stimulus frequency emission is the largest. However, it's not very easy to make a clinically robust test to detect that. And so what we use is gaps between the stimulus in order to measure a stimulus frequency response, and that is the transient emission. And finally, we use another phenomena related to amplification, which is that it tends to slightly distort the signal, and we can look for the presence of frequencies that were not in the stimuli as an indicator of that active amplification process. So the clinical focus is generally on DPs and TEs, mainly because they are the most robust and reliable ways to measure this phenomena of amplification and active processes in the cochlea. It's the most reliable method to observe the outer hair cell function.